Thanks, everybody, for coming today. I am pleased to introduce Abuzar Mahmood. He received a Bachelor's of Science in Physics from University of Missouri and a Master's and PhD in Neuroscience from Brandeis. And he's currently a postdoc in Don Katz's lab. And he studies sensory processing, such as taste with electrophysiological and statistical modeling. His dissertation included brain regions involved in taste processing and found they may form an attractor network. And we look forward to hearing more with research.
Thank you, Nate. I'm really excited to share with all of you my work looking at, let me get rid of that thing first, the interaction between two of the brain regions we consider to be really important in taste processing. Wait, does someone know how to get rid of this?
You drag the bar.
Somewhere, okay. Goodbye. Okay. Two of the brain regions which are very important for taste processing, the Gustatory Cortex and the Basolateral Amygdala. I want to start by thanking Sreshta and Nate for inviting me here and for organizing. A couple caveats before I start. So I do a lot of data analysis. So there will be descriptions of the data analysis here, but given the people in the room, I apologize that there won't be too much discussion of that. I'll be happy to discuss more in detail later, but this will be more a neuroscience and biology heavy talk. The other caveat I want to throw in is I'll use the word information a lot. And, for those of you who have met Paul Miller, throughout my PhD, he kept telling me, you're using it wrong. And he's right. I don't mean it in the Shannon information kind of sense. I'm using it very colloquially, in terms of like encoding. So I hope you will indulge me in the use of that. Right, so this talk is a lot about kind of where my project came from and the context it's set in. And so there'll be a lot of background on what the lab has done in the past, and then at the end, I'll talk a bit about what I've included, but what I've added to the lab's work. But the reason it's set up that way is because there's a lot of kind of niche neuroscience-y things that the lab does that is not common. And so that's my attempt to first give a foundation of where my work is coming from. So how we think of taste processing in the lab, the philosophy behind it is that the job of the brain is to make sure you stay alive, right? It's to make sure that you ingest nutrients and that you avoid toxins. Nutrients usually come from foods which are salty or sweet. And then toxins usually are associated with things that taste bitter or in high doses sour. And when we give rats these tastings, understandably, their behavioral output looks very different. So this rat, which has received either of sweet or salty, it's a very subtle movement, but I hope you can see it's kind of swishing its mouth. It's licking inside its mouth, gathering up all the fluid it's been given in preparation to swallow it. Instead, if we give the animal a bitter or a sour taste, it makes these very stereotyped mouth opening movements where it's kind of rolling its tongue forward in an attempt to spit out the taste. And they do other things like literally try to claw it out of their mouth, but the behavior we can most easily record is gaping. And so we'll talk more about that later on in the next few slides. Okay. So at this point, you might ask me, why is it relevant? Why is it useful to study these in rats? Because there's all kinds of organisms, they all have all kinds of idiosyncratic behaviors, different neural systems, and why is it useful to study this in rats? And so my answer to that is that you'll have to believe me that the systems involved in processing taste across species are reasonably strongly conserved, and the behaviors are also reasonably strongly conserved. For example, if we take my boss's son here, responding to a recently ingested sour taste, sticking his tongue out, trying to get rid of it as a gape, right? But this goes all the way back to amphibians, where these hungry frogs here will try to eat this critter that's walking by. realize very quickly they've made a horrible mistake and then stick their tongue out to try to get rid of it. I just want to make sure people can see the slides fine. Should I try to turn off the lights? Maybe. Board lights.
Yeah.
Better? Okay, great. Right, so the job of the system across species seems to be its job and its output seems to be reasonably conserved. So we have reason to study this in rats. And as we're studying this, this process for taste decision making, we need to keep in mind that biological systems, unlike artificial neural networks that we're used to, make this decision making, make this classification in the process of time. There's biological timescales involved with the brain. And the other part is that the brain is not a random network. There's specialized connections, there is specialized regions. Many parts of the brain can be described well using randomly connected networks, but a lot of it is pretty specialized. And so keeping both of these in mind, we think of taste processing as something that unfolds through time and across multiple scales of the network, multiple scales of the brain. And so what we believe and what I hope to show you today is that time matters in neural processing. We'll start by looking at the response of single neurons in the cortex. And we find that this work that was basically what got Don Katz's faculty position here shows that the response of single neurons from the gustatory cortex evolves in a meaningful way across the 1 to 1.5 seconds following stimulus delivery. That different parts of the response carry different meaning for the unfolding taste response. And for those of you who were at my criticality talk for Comp Neuro Journal Club a few weeks ago, you'll remember that I showed some evidence for how brain behavior, or the properties of activity in the brain, is critical. And if you don't want to go as far as claiming criticality, it's at least multi-scale, spatially and temporally. And so it makes sense that the kind of dynamics that we see at the single neuron level, we also observe at the multi-neuron level or the ensemble level. And we find the neural ensembles work together to process taste. I'll show you some data on a single trial basis, which shows that these neural ensembles work together with strong nonlinear transitions that we believe drive behavior. And taking the multi-scale part all the way up to brain regions, I'll show you my work looking at the interaction between the gustatory cortex and the basolateral amygdala, which we think couple into a single processing unit for the purposes of taste processing. And we believe that the interactions between these brain regions drive the dynamics that we see. At any point, if people have questions, feel free to stop me. I prefer we all move along with clarifications done than someone not understanding what I'm saying. So when I joined the lab back in 2018, there was not a single paper studying interaction dynamics in any part of the brain circuit, in any part of the taste circuit. Right? There were papers from other sensory modalities, which were looking at dynamic interactions that we could draw inspiration from, but nothing in taste research itself. And I like to blame this on what I call the canonical pathways of taste information flow in the brain, where there's been this idea for some 2 odd decades that the taste circuit is a purely feed-forward neural network. So when a tastant hits the tongue, signals come up from the tongue via the cranial nerves into the brainstem, shown in green, and there it splits into 2 pathways: the limbic pathway, which contains the amygdala, and the thalamic pathway, both eventually converging onto GC, the gustatory cortex. Now, you don't have to remember most of these brain regions. The ones I'm interested in, I'll bring them up later. But if we take a closer look, we find that the connectivity for the circuit looks more like this. And we also know that, we also know that each of the brain regions shown here are simultaneously active after a stimulus has been given to the rat. So it makes sense that all these simultaneously active regions are influencing one another, are coordinating using these reciprocal interactions. And that rather than one brain region processing something at any given time and then passing it on to the other, that all of them are moving ahead in concert in a coordinated fashion to process the taste, to process the decision, and to generate the behavioral output, right? So it's not, no brain region is processing in isolation. We think that all of them are working together. And so my project to try and start parsing these interactions focuses on the dyad formed by the basolateral amygdala and the gustatory cortex, both of which we know to be strongly reciprocally connected. And they're both regions important for taste processing, taste-associated learning, and decision. So we could have started with any pair of brain regions, but these are the two that the lab has primarily studied over the past 20 years. And so that's where we began. So a quick overview of where we'll go from here. I'll talk at a reasonable length about the properties of GC and BLA taste responses individually, again, to give context for where I'm headed. And then I'll talk about what I've learned about, or what I've been able to learn about the GC amygdala interaction in two parts. And the reason it's broken down into two parts is because we put a first paper together, and then after that we were like, okay, we can do a little bit more, we can make more sense out of this. And so the second part was follow-up project based off of that. So far so good. I'll just start a timer to make sure I'm not running over time. Okay, before we go on, I just want to make sure people are familiar with what a raster is and what PSTHs are. Anybody would like me to? Oh, perfect.
The raster.
Yeah, so even if you give the same stimulus and record from the system, neurons, they're somewhat stochastic, right? So you don't get the same response every time. And so generally what people do to figure out what's signal and what's noise is give the stimulus multiple times. This is 40 times. In all the slides that will come next for my own data, we give it 30 times. And then you average it. And so each, you're familiar with how neurons spike, right? Like action potentials. Yeah. So if you think of a neuron's activity as just a binary response, you can mark it as a point process. We can just say this is when the neuron was active across multiple trials. And that allows us to get kind of like a average look at the properties, the response properties of the neuron. Right? And if you collapse across all of them, then you get the PSTH that you're probably more used to seeing.
So the first box, that's a raster.
Yes.
So over your 40 trials, on average, it spikes somewhere around 1 time. It's like most of them are, or like closer to 1. Right.
Yeah, so this is where, generally what happens if you give a stimulus, the brain kind of lights up. Right. And so this specific neuron shows most of its activity surrounding after the stimulus delivery time. And most of the dots, so each dot is an action potential. And this neuron seems to become very active right after the stimulus.
So is the 0, 1, 2, 3 time?
Oh, yes, it's time, sorry. All the way down here. But yeah, it's time from the start of the recording. Good. Okay. So let's start by looking at what we know about the response of gustatory cortex neurons. So here is a PSTH from a single neuron recorded from the gustatory cortex in response to 4 different stimulants. Sodium chloride shown in blue is salty, and sucrose shown in orange is sweet. These are both palatable tastants. And then citric acid in green is sour, and we give it in high enough concentration that the animals don't like it. And then quinine shown in red is bitter. This is the trial average response aligned to the time of stimulus delivery, which we can control with very high accuracy. And so I hope looking at this figure, you can see that the evolution of the response of this one single neuron can be roughly broken down into 3 time periods. The first of them we call the first detection, which lasts from about 0 to 250 milliseconds following stimulus delivery. We consider this to be a somatosensory response. There's very little taste-specific information present in the system at this time. So you can see that even this early, there is some separation. But if you try training a classifier to separate out the taste, it'll give you, very poor accuracy at this time point. So it's mostly somatosensory. Just a response to something hitting the tongue. Taste-specific information first begins to appear in the responsive gustatory cortex neurons after about 250 milliseconds or so, 250 to 800 milliseconds, in a period which we call identity. All of these are very creative names. And so at this time period, basically, if you train a classifier on this time period, it would actually do well in separating out what stimulus was given. But arguably, the most interesting part of the response comes after about 800 milliseconds or so, when we find that the response of neurons in the gustatory cortex begins to separate by the hedonic value or the palatability of the tastes that were given. So sodium chloride and sucrose, shown in blue and orange, separate to one side, and citric acid and quinine separate to the other. So these neurons are encoding the palatability of the taste that was given. Now, intuitively, it makes sense what's palatable, what's unpalatable. As scientists, you should ask me, how do we know that rats perceive these tastes in the same way that we do? And the answer is that you can do a behavioral assay where you can let the rat lick or try to consume each of these tastes as many times, as much as it wants, for, in this case, 15 seconds or so. And we find that, as expected, rats drink sucrose and sodium chloride the most, and then citric acid and quinine much less. So we can try to see how much of this ranking of what we call this the canonical palatability ranking, how much of this is represented in the neural activity by simply doing a Spearman correlation with the PSTHs of the neurons. And that gives us something like this, where the crosses in this black line denote when the correlation became significant. And we can see it happens only after about 750 milliseconds or so. So this time period, again, very creatively, we call the palatability epoch. And so the point of this is to show that there's different, quote unquote, pieces of information in the response of GC, gustatory cortical neurons. But I'll draw your attention to the fact that in this trial average PSTH, what appears to be happening is that the activity of this neuron seems to be changing on the order of hundreds of milliseconds, very smooth. And that's seen across PSTHs for many, many systems. But things actually look pretty different when we look at the response of GC neurons on a single trial basis. So to illustrate what I mean, here I'm showing the response of 10 simultaneously recorded neurons from the gustatory cortex to quinine. And I'll draw your attention to the time of this red line. And what I hope you can see is that rather than changes happening smoothly, instead what we find is that the activity of these 5 neurons highlighted in green shows a sudden increase across the line, whereas the activity of these 2 neurons highlighted in red shows a sudden decrease across the same line. So rather than a gradual change, instead, what we believe is happening is a sharp transition from one pattern of population activity to another. And we can detect these kind of state shifts in ensemble activity using things like hidden Markov models or change point models. And so here I'm showing you a single illustrative example for what one of these transitions could look like, but we could apply these models to the entire 1.5 to 2 second of post-stimulus activity that we record from neurons in the gustatory cortex. And so the whole thing is a response to a single tasting. And using a hidden Markov model, we found where states and state transitions occur in this response. And what we find when we fit these models is that the constitution of individual states, the pattern that describes any individual state, is very consistent from one trial to the next. So if you looked at any one trial and I was able to tell you what state the system was in, you would expect very reliably that the neurons fire according to these patterns. But and the other part is that the sequence of these states is also very consistent. So the system always goes through state 1, 2, 3, 4. What is not consistent is when these states occur. So these are multiple trials in response to the same stimulus. And if I can draw your attention to this green state, we can see that from one trial to the next, the timing of its onset is highly variable, and the timing of its duration is highly variable. So it seems like that the system goes through states, but their duration is not consistent. Okay. So this was all a lot of fun, a lot of characterization of population activity We made some nice plots. We showed variability in the data. The question becomes, why is this relevant? Why should we care? And that's the same question the authors of this paper asked. What do these dynamics tell us about processing happening in the system? How is this related to the generation of behavior? And so one thing I didn't tell you is that gaping, the generation of the response to the aversive behavior, happens around 1 second on average following stimulus delivery, but its timing from any one child to the next shows some variability as well. And so to try and understand how these neural dynamics relate to the generation of behavior, what the authors tried to do is focus on the relationship between the transition to the sport state, which seems to come on around the same time of the onset of gaping, and see whether there is a relationship there or not. So just to remind you what kind of a gape looks like, the authors did a pretty straightforward analysis where they simply compared on any given child the time of the neural transition to the latency of the onset of gaping to quinine. And if data falls above the identity line, then the neural transition happened first. And if it falls below, then the behavior occurred first, right? And the authors found that very reliably. And obviously there's some spread given noise in the data and given noise in inference, but pretty reliably, the neural transition precedes the onset of gaping by about 300 milliseconds or so. And so this tells us that there are changes happening in neural activity in the gustatory cortex that will predict the onset of behavior. And this was our first, or one of the more concrete first implications for the involvement of the gustatory cortex, not only in the decision-making part of the taste response, but also in the generation of behavior part. But this is still phenomenology. This is still correlation. To try to causally show that GC is relevant, it's responsible for the initiation of gapes, took a few more years. And so work done by graduate student Narendra Mukherjee tried to answer this question, where what Narendra did is injected a virus in the gustatory cortex, which allowed him to very precisely optically inhibit activity of the neurons in the gustatory cortex. And so this is a spatially localized perturbation. We can only perturb where the virus infected neurons in the gustatory cortex, and it's also temporally very precise. We can control it by implanting an optical fiber in the gustatory cortex and controlling very precisely when the light is administered. And so Narendra implanted this optical fiber and also electrodes at the same time to record activity from neurons in the gustatory cortex. And instead of doing one big perturbation for the entire duration of time I've been showing you so far, the 2 seconds following stimulus delivery that we seem to be so interested in, he did 3 smaller perturbations, each of 500 milliseconds, with the idea being that if GC is involved in the decision-making and the generation of behavior related to patients, then perturbations before or around the behavior will have an impact on the generation of behavior, and the perturbation that comes after shouldn't really do anything.
Can you clarify what does the light do, or how does light influence the behavior of the neuron?
So the virus encodes something, a channel that gets embedded in the neuron's cell membrane, and it's light activated. So when we implant this optical fiber and shine the light on that neuron, I think it's a chloride pump. So it pulls chloride in, hyperpolarizes the neuron, and prevents it from spiking. And we can do that for large areas of the gustatory cortex, right? We can't reliably cover entire gustatory cortex because brain regions are 1 amorphous and you need to put in a lot of optical fibers, but we can do enough to, as you'll see, have an effect. Okay, any other questions? Okay, so Narendra did these perturbations and tried to see how they affected the onset time of the behavioral response from taste delivery. And so as a reminder, without a perturbation, the onset of gaping happens around 900 milliseconds. It changes a little depending on what animals you have, anywhere from 800 to 1200. is expected, in this case 900. And he found what we expected, that the early and the middle perturbations, those which reliably happen before the transition into the palatability state, before the generation of behavior, were the only ones that significantly delayed the generation of behavior. And they did so by 500 milliseconds, which is exactly the amount of time the perturbation was on. So in a way, perturbing GC prior to the generation of behavior basically prevents the system from generating a behavioral output. Now, we're not entirely sure exactly how it does it, but what this tells us is causally, the gustatory cortex is involved in the generation of the gaping response. Okay, so now that we know GC is causally involved in decision-making and the generation of behavior, we need to keep in mind that GC is not an island, right? There's all of these dense connections, recurrent connections between GC and the rest of the taste circuit, and that nonlinear dynamics are hard to explain without loops. And why would you try to do that when there are already so many loops present? So remembering that GC is strongly connected to the basolateral amygdala motivates our question of, what are the interaction dynamics between these two brain regions? And before we get to the interactions themselves, we'll go through a little bit of what we know about the basolateral amygdala, or response in the basolateral amygdala themselves. So here is the response of a single neuron from the same four stimuli. And we know that these neurons aren't doing exactly the same thing that the gustatory cortex is doing at any given time, but the dynamics, the changes we see in this neuron's response line up very well with what we see on a trial average basis in the gustatory cortex. So namely, the palatability epoch or palatability information is already present in the basolateral amygdala at the same time where only identity information is present in the cortex. Now, I haven't included a basically neuron average palatability time course for GC, but you can take my word that at this point there is very little palatability correlation present in GC, but it's already present in the basolateral amygdala, right? So there is still an alignment in their dynamics, even though they're not encoding the same thing. And so if we think BLA and GC are strongly influencing one another, they're strongly interacting, then the standard way to look at or try to assess this interaction is by looking at coordination between the activity of these two brain regions. So this leads us to our first set of predictions. We think that the basolateral amygdala, much like the gustatory cortex, activity in the basolateral amygdala will evolve as a sequence of states. If that hasn't been shown before, we expected because BLA and GC are strongly connected. Would be very interesting if that weren't the case, but a priori, that's what we expect, right? But we don't just expect that BLA and GC will show similar statistics of population activity. What we actually expect is that BLA and GC activity will be coordinated. The state transitions will be strongly coordinated. And I'll come back to why we're so interested in the second transition in a few later slides. But for now, let's see what we can say about BLA population activity. So if one, and by one I mean me, chooses to go look for population activity in the basolateral amygdala, one is indeed rewarded. Here is the response of a single BLA neuron aligned to stimulus onset. In this PSTH, you can see that the activity of the neuron seems to be changing slowly, gradually, on the order of hundreds of milliseconds, but that doesn't seem to be the case in the raster above. In the raster above, we can see that there is a sharp uptick in the neuron's firing rate, but this is happening at variable time points across trials. And so when you average them, you artificially get this gradual increase. But we can use the same models that we've been using, hidden Markov models or change point models, to find these changes in the neuron's activity and align them to the timing of the transition, so that when we average them now, we're able to recapitulate the sharp increase in the neuron's activity in the trial average activity, right? So I showed you my very pretty cherry-picked neuron for this example. Do we see this in population activity? Yes, we do. These are 8 simultaneously recorded neurons from the basolateral amygdala responding to a single tastant. Inferred states are shown by different colors. I'll draw your attention to the transition from state 2 to state 3. where the 3 neurons underlying in green show a sudden increase in their activity across this transition, and the 2 neurons underlined in red show a sudden decrease in their activity across this transition. Right? And furthermore, we can ask how many states, models with how many states best describe population activity in the basolateral amygdala for this time period of 2000 milliseconds that we're interested in. And we find very reliably the answer is model with 4 states. And this is a good sign because you also need model with 4 states to best describe the evolution of population activity in the gustatory cortex, right? So far, things seem to be lining up, right? We're a little happy, but we still have a bit more work to do. Because up until this point, no one had shown population states in the basolateral amygdala. And in the previous paper for GC, Don had gotten a lot of pushback about states being present in the GC, so much so that it took 3 years to publish the paper. We wanted to make sure that before we put this outrageous claim of population states present in BLA into writing, we can back that claim up. So there's multiple ways we could have gone about this. You could have done a direct modeling comparison. Similar to what had been shown for GC, decided to go a different route. The logic is that if this complicated model, whose only job is to pull out state transitions from data, is being fit to, let's say, random noise, right? It will give you something that looks like state transitions. It will pull out something that seems like signal from random noise. So let's say our data actually had state structure, like this is just some cartoon data, but the point is to show that there is these specific states with very specific population patterns that are variable from one child to the next, and that neurons transition from one state to the next collectively. Right? If this specific structure is present within the data, then the model's fit should get progressively worse as we mess with these assumptions, as we start destroying the coherence and the state structure present in our actual data. Right? We don't know whether it's present, we're trying to confirm, but if we do manipulations that mess with that structure, the model should fit worse and worse. And so we can start by destroying population coordination, such that we can take the activity of single neurons and shuffle them across trials. So now in the new data set, the activity of neuron 1 on trial 1 is paired with the activity of neuron 2 from trial 10. So on any given trial in this shuffled data set, individual neurons do have sharp transitions in their activity, but the coordination across the population is lost. So the model should have a harder time fitting. We can go a step further and just completely destroy all sharp changes. We can shuffle single spikes for any given neuron across trials. And that kind of enforces any single trial to more closely follow the trial averaged PSTH, which, as we know, only shows smooth changes on the order of hundreds of milliseconds, right? So if there aren't any sharp transitions, then the model should just have a really hard time fitting. And so that's exactly what we see. These change point models were fit using a Bayesian modeling package and using a metric called the evidence lower bound, which is just, it's a penalized, I'm at loss for a word. It's a parameter penalized optimization metric. So it accounts for, it penalizes models with larger number of parameters more. And we find that comparing the models using this metric, indeed, the shuffled data sets showed much poorer fits compared to our actual data. So this gives us more confidence that the kind of nonlinear dynamics that we're trying to describe in our GC, in our BLA activity actually are present in the data. Okay. So I got very excited. I put a little tick mark next to this prediction. I went to Don, did a little dance, and then he was like, our question is actually to ask how BLA and GC transition are coordinated, right? The first prediction was in service of the second. Any questions at this point? Anything I can clarify? Okay. So if you recall, the second transition is the one that precedes the palatability state, and the gustatory cortex is the one that precedes the onset of behavior. And we have reason to believe that something special is happening between the gustatory cortex and the basolateral amygdala interaction around the second transition. So I'm going to throw up the schematic once again, and looking at it more closely, given the fact that BLA shows palatability information before the gustatory cortex, it might suggest to us that palatability representation is transferred from BLA to GC at the time of the second transition. And an interpretation like this is supported by previous perturbation data from the lab, which wholesale inhibited activity in the basolateral amygdala using a pharmacological agent. And this perturbation supposedly blocked this transfer of palatability. And what we observed was a decrement in palatability encoding present in the gustatory cortex. But this is a very coarse perturbation, right? BLA is also connected to many other brain regions. If you knock out a whole brain region, it's going to have higher order effects that we can't explain. So to do a more precise perturbation, both temporally and spatially, the senior postdoc in the lab, my colleague, friend, and brother from another mother, Jianyu Lin, decided to infect the basolateral amygdala with ARCH-T, the optical silencer. And what this did was it not only allowed expression of the silencing in the cell bodies of neurons in the basolateral amygdala, but also the projections from the basolateral amygdala going into the gustatory cortex. And then what he did next was he didn't implant an optical fiber in BLA, but instead put it in the gustatory cortex, which allowed him to very specifically block the input coming from the basolateral amygdala to the gustatory cortex without affecting activity of cell bodies in the basolateral amygdala themselves. So this specifically allowed him to cut the connection or the input from BLA to GC without affecting the circuit at large. And he also implanted electrodes to observe what was happening in the gustatory cortex under this perturbation. And what he found was that when this connection was cut or optically perturbed, there was a net decrement in the number of GC neurons encoding palatability information, right? Something we expected. But the purpose of this more temporally and spatially precise perturbation was to get a clear answer about what is happening between GC and BLA around the second transition. And to study this, Jianyu looked at the onset of the palatability response in the gustatory cortex around the time of the second transition. And so this is a peri-transition time plot. And as expected, Jianyu found that when the connection is intact, there is a sudden sharp rise in the palatability response in the gustatory cortex. But when the connection is perturbed, this rise takes much longer. And so this tells us that really something is happening, something special is happening because of BLA input in the gustatory cortex, that BLA input is needed for sharp coherent transitions present in the gustatory cortex. But what's been missing up to this point is a direct assessment of the interaction dynamics between the basolateral amygdala and the gustatory cortex. Just making sure I'm good on time. What, like 5-10 minutes more?
Yeah, at least.
Okay.
Problem.
Cool. So to answer the question of interaction dynamics. Oh, okay. How long has that been up there? Okay. Goodbye. Okay. So to try to study interaction dynamics, for my project, I implanted electrodes in both the basolateral amygdala and the gustatory cortex, something I will never do again. I'm not a great experimentalist. I'm much better at analysis. And I fit models, the change plane models, to neural ensembles recorded simultaneously from BLA and GC independently, right? And once these models were fit to the ensembles, I compared how strongly aligned are the neural transitions inferred by the model. Okay, and so here I'm showing you raster plots for a single child. Each row is a different neuron split by GC and BLA. And I've highlighted inferred states with the different colors. And I'll draw your attention to one specific transition denoted by these yellow rectangle blocks, right? So looking at coordination of state transitions is a particularly stringent measure of coordination between brain regions, because the timing of these transitions is variable from one trial to the next. And so it becomes very unlikely that you'll see this kind of coordination by random chance, right? So this one trial showed coordination between BLA and GC for this transition. If we plot in a few more pretty trials, we see that even though the timing of this transition moves from one trial to the next, it's aligned between BLA and GC, right? These are representative figures. What do we see when we compile our data across all transitions and across our entire data set? So here we're looking at the percentage of data sets with high BLA and GC correlations. And we find for the first transition, the one from detection to the case-specific states in both BLA and GC, that transition isn't strongly coordinated between the two regions. But the following two transitions are, and so this result bears out our expectation that something special is happening between BLA and GC around the time of the second transition, that the two regions are coordinating to generate this transition. But looking at coordination transitions only gives us a snapshot of the coordination between these brain regions. What we expect is likely happening is that the basolateral amygdala, given the studies I showed you earlier, given Jinyu's work, is basically pushing the gustatory cortex to the second transition to the palatability state. And so what more can we do to try and understand this, right? So another caveat of this analysis, looking at state transition coordination, is that it's a symmetric measure, right? We reasonably cannot expect that the influence going from BLA to GC and from GC to BLA is symmetric, simply because the physical connections between the two regions in each direction are different. So it would make sense that the influence they're exerting on one another also has to be different. The other part is that while we showed in the last slide that state transitions are coordinated between the two regions, they don't have to be synchronous, right? There could still be a time lag between one of them. And a time lag in things like state transitions or other kinds of measures of coordination of activity is taken to mean that one region is driving the other, right? So if, let's say, BLA transitioned first and GC reliably followed, it would suggest that BLA is pushing the gustatory cortex to perform that transition rather than the two regions interacting together, right? So we need to do a few more things. We need to, if we're asserting that the two regions are coordinated, then what we want to do is try and see whether they move together in lockstep, whether there's any lag between them or not. We want to break down the directional influences going in each direction, because so far we only have a symmetric measure showing connection strength. And we want to do this in a more time-resolved way, right? We want to know what is happening before the second transition, how influence from BLA is changing over time. Okay, so new set of predictions. Rather than saying the second transition is coordinated, we'll actually say that it's synchronous, right? It doesn't really have a time lag. Because we believe that BLA will be driving GC into the second transition, after which, as was shown by Narendra's work, GC emits the behavioral command. We want to break down directional influences in a time-resolved way. But this is all big picture ensemble region level analysis. What about the single neurons carrying out this interaction? We expect that the neurons in GC and BLA involved in the communication between the two regions are the neurons most important for the task relevant circuit, right? And so because they're important for BLA-GC communication, because they're important for BLA and GC coming to this decision, they will likely have the strongest taste encoding. Okay, I will speed up just a little bit. So if we look at transitions between BLA and GC, negative means GC leads, positive means BLA leads. For the first transition, BLA has about 100 millisecond lead on the gustatory cortex. And if we didn't know that this transition wasn't coordinated between BLA and GC, we would have assumed that BLA is pushing GC to this transition. But more so, what it can probably mean is that at this point, there's still separate circuits, functionally separate circuits, and are being driven independently. Very interestingly, the lags for the following two transitions, which are strongly coordinated between BLA and GC, have on average a undetectable difference from zero lag. I was told by a reviewer to say that. Right, we can't prove a null, but we can say that on average there isn't really a lag between them. And so this supports our idea that after an initial transient, the two regions become coordinated in response to a stimulus delivered and carry out taste processing collectively, right? But what is happening with the directional influences? So we chose to look at directional influences using something called Granger causality. People know what Granger causality is? No? So it's basically you fit autoregressive models for both the time series that you have, and the claim would be that if you're trying to predict the future of GC activity using the history of GC itself, if you add the history of BLA and it does better than just accounting for the history of GC, then that would suggest that input from BLA is important to predict the future of the gustatory cortex, right? And so it's not technically causality, it's called Granger causality, right? That's a caveat. And some smart people figured out how to break this down by specific frequencies. I won't go into that, but what we find when we look at the BLA to GC influence, and here the heat of the color tells us how many data sets showed a significant interaction around this time. We find that the bulk of the BLA to GC input lies exactly in this second state. Right, so the push from BLA to GC seems like is happening during the second state, leading up to the second transition. Whereas if we look at the GC to BLA influence, we see this burst after the end of the BLA to GC input, and we're interpreting this currently as the behavioral output sent by the gustatory cortex, not necessarily specifically to BLA, but to the rest of the taste circuit to trigger that transition to begin the onset of the gaping response or whatever behavioral response, right? And so we're not only interested in the temporal dynamics of this interaction, we're also interested in the properties of the neurons underlying this interaction. And so we can trade the temporal resolution that we get from Granger causality to try and study the links between pairs of single neurons between GC and BLA. And we do this using something called Poisson generalized linear modeling, which is a somewhat conservative way for us to find out that above and beyond modeling input history, the history of the neuron itself, what does the history of other simultaneous recorded neurons contribute to the neuron we're currently focusing on? So we fit GLMs to every single neuron we recorded, and the model tells us every time it thinks there's a significant input from the history of other neurons. And this is functional connectivity, right? The concrete way to do this would have been to somehow label the projection neurons from GC to BLA and record from them. But again, because I'm not a great experimentalist, I decided to bootstrap the data we'd already recorded and try to infer functional connections. And so every time we get a significant coupling filter or a significant link between history of other neurons and the neurons we're looking at, we said that is a connection, right? We can tabulate these connections for the gustatory cortex, and we find that there is roughly an even split in neurons influenced by BLA and neurons sending output to BLA. And there's a healthy overlap between these two inferred subpopulations. But the goal was to look at functional properties of the neurons denoted by these inferred connections. And we do that by looking at the discriminability and the palatability information encoded by the response of these neurons. So discriminability is just, again, we apply an ANOVA, and it's the effect size of the ANOVA. How different is the response of the neuron across tastes? And palatability is just the palatability correlation. So we find that neurons influenced by BLA show much higher levels of both discriminability and palatability compared to neurons sending output to BLA. But the neurons which have the bidirectional connections, arguably the ones which are most strongly embedded in BLA-GC interaction, are the ones which show highest levels of both. And for our convenience, we can combine both palatability and discriminability into tastiness, which is basically just the hypotenuse if you plot discriminability against palatability. would just be the the L2 norm of the vector. And we find that for the overlap population, this is significantly higher than the neurons receiving input simply from BLA. One more quick note. The reason we find this particularly interesting is because all of these neurons have the same inferred putative cell type. We didn't actually go in and do a genetic screen, but we can look at the shape of the waveforms that are recorded and see whether these are pyramidal or interneurons. And generally, people expect these kind of functional differences to be broken down along the lines of cell type. But all of these were pyramidal neurons, putative pyramidal neurons. So it seems like more than cell type, the functional properties of these neurons are determined by the connections that they have. Okay, hopefully I was able to show you that GC taste responses reflect processing across successive epochs. Ensemble analysis of single trials reveals coding to emerge suddenly across the ensemble, and that these transitions drive behavior, likely by modulating a brainstem CPG. That's not my project. We have CPG experts here. If you want to talk to someone in the lab, I can put you in touch with them. But we think GC just sends an output to trigger the CPG and then forgets about it. We think the mechanism of construction of these transitions involves BLA and GC coupling into a single functional unit. We think, obviously, it's probably not just GC and BLA, the rest of the taste circuit is involved in that too, but this is how far we've gotten in looking at coordination. And so with that, I'd like to thank my boss, Dr. Donald Katz, our fearless leader. I've shown you data from a lot of previous graduate students and postdocs in the lab. In particular, I want to give a shout out to Jessica Steindler and Hannah Germain, two of my mentees who immensely helped me collect data and perform analysis, without whom much of this work would not be possible. I want to thank Don's collaborators, in particular Paul Miller. And this statement has been filtered by Don because apparently it scandalized Paul at some point. But Don says Paul does the math because Don does not know how to count. And our funding sources. And thank you all for attending and for your attention. Happy to take any questions. I also love speculating.
So, good question. What are your intended next steps with this project in terms of analysis?
Yeah, so my dream would be to have an analysis lab where I never have to collect data again, but it's a little harder to get funding for that. So there's a couple ways we can think about going. One is to incorporate more brain regions. Right? In the beginning, if you remember, I mentioned that from the brainstem, there is a split along the limbic pathway and the thalamic pathway. We know that input from each of these pathways converging onto the gustatory cortex do different things. But no one's tried looking at different in what way, right? We have inclinations from people inhibiting thalamus, inhibiting BLA, but there isn't a synthesis. No one's done, a triple region recording and trying to figure out where they're redundant and where they're not. The other thing would be to look at GC amygdala interaction in cases of learning. In cases of maladaptive learning, GC and amygdala are, there may be there are 2 of 3 brain regions which are involved in almost all major psychiatric disorders, anywhere from schizophrenia, depression, PTSD, anxiety, eating disorders. Insula and amygdala involved in that, which makes sense because insula is basically a hub of sensory information, and because it receives strong input from both prefrontal cortex and the amygdala, there is a lot of emotional information that it controls. Even if you can argue that it doesn't generate it, it's basically a mediator for a lot of emotional information. So those are 2 angles I'm looking at. We'll see if I can get any money for that.
You talked about the connection between GC and if you block, I think you showed data that I'm not quite sure if I follow that on GC block, but you just have that BLA, you block it. How does that affect the behavior of the patient?
So I think by and large, it actually doesn't affect generation of behavior. Right, I think it mostly affects learning, but it doesn't affect generation of behavior in a single child. It's one of those things where like, is it behaving differently in an intact circuit than it would in a compromised circuit? Like, does the rest, if you inhibit BLA, does the rest of the system pick up? It's hard to say whether BLA has a well-defined bottleneck role, or is it simply a modulating influence? But we know it's doing important things. It doesn't directly control behavior. If you don't account for learning.
Yeah, I have a question. Can you go back to, your second, the last slide?
This guy?
No, after this, right before your thing, side thanking people.
Oh, I see.
Yeah, so you mentioned that the third one was possibly modulating the central pattern generator.
Mhm.
And I noticed that some of the states, you associated them with rhythmic activity, potentially, like certain rhythm bands seem to be particularly active. Yeah, like this, and I don't know what slide it was earlier, but I also am studying rhythmic activity, and one thing that was interesting is these state changes. I would wonder if you studied like if some neurons are influencing the rest of the neurons to cause state changes, because when you show the neuron activity, you
Right.
show like all the neurons kind of changing in a state at the same time.
Right,
But
that's a good question. So we've been operating under the assumption of a model Paul Miller and Don put together back in 2010, 2012, which suggests that the transitions are noise driven. And so there isn't particularly a source or sink. And so according to that model, you wouldn't expect consistently a set of neurons to drive the transition. I think that's wrong. So we haven't looked at whether, to put your question a slightly different way, whether there's a sequence of neurons as the transition happens. That's something I've wanted to look at, but I haven't had enough time. But I think your intuition is right, that there are specific mechanisms for each of these transitions. I don't think they're noise driven. Noise probably has a role, but I don't think it's driving them. So we haven't quite figured out what might be driving them. Yeah, there's some really interesting work. I don't know if you've heard of global workspace theory. But in visual processing, they posit that, so they basically see the same states we do, but in visual processing, right? A lot of people don't like the idea of these states, but if you go looking for them, they're everywhere. They're in every single sensory modality. And so the idea is that initially, when a stimulus is given, there's a feed-forward pass. And then once the brain regions along those feed-forward paths become activated, they start interacting via their feedback connections, and the whole circuit starts working in kind of an internally driven reciprocal fashion. And so, that is definitely not a noise-driven mechanism, but I've only come across that idea fairly recently. We haven't worked out exactly how that can be linked to the mechanisms of the states that we see.
yeah, because that would fit with the different brain regions contributing.
Right, so that's, and there's it also very well explains the effects of the perturbations we see, because the first state, according to global workspace theory, is stimulus driven, which means if you perturb the first state, there's still stimulus input coming in to override your perturbation. And so the system can continue despite the perturbation to an extent, right? Which is, I didn't go too much into Narendra's data, but that to an extent is what we see. The second state, which is internally driven, has the strongest impact, it suffers the most from a perturbation. So that's expected from global workspace theory, and that's what we see.
So, yeah, 'cause that was. And some of the work I'm looking at is kind of addressing some of the things you're saying, like, I'm. Trying to interpret functional connectivity
Uh-huh.
sometimes compared to actually knowing the structural connectivity
Mhm.
based on the recording data, so.
Yeah.
Trying to find ways to find which neurons are influencing other neurons.
Right.
you know?
It's challenging. But yeah, I'd be happy to talk later if
Yeah.
you're interested.
Yeah, I'd be very interested. Thank you.
Yeah, thank you.
This is, I guess it's kind of related to when you're talking about like learning.
Mhm.
But if I understand correctly how these experiments are done, at least a lot of it, is you're doing many trials
Mhm.
in a row, so you're like giving the same.
So not the same. I didn't go into too much detail of the experimental setup, but the tastes are delivered in a pseudo randomized order. And at least for my experiments, I made sure that the animals never got the same taste twice. But yeah, it's beyond that. It's 30 trials of the same tastings.
I was wondering if you would, do you see these sort of transition
Mhm.
times like drift or anything as you apply the stimulus? Sort of like it's the first time
Yeah.
the mouse perceives the taste, does it take longer to react?
So yeah, that's very perceptive. The system does kind of like equilibrate after a few trials. The first few trials, the response of the network is different. You can say like the first 5 trials of every tasting are different from the remaining 25. And you could argue just from a Hebbian perspective, right, that we're giving these animals a novel stimulus, which will cause learning. And eventually the system will equilibrate to the new stimuli coming in. As to whether processing is different, I think the jury's still out, probably, because there's learning happening. And the animal, even if you think in terms of top-down influences, the animal is now expecting taste to keep coming in for a while. We haven't looked at it explicitly, but I'm sure the timing of the behavior changes. Whether that's meaningful is a different question, but yeah, it definitely changes. Both the dynamics and the generation of the behavior changes.
We have 2 questions. One that's something that is, does that make it the situation where, like, let's say the first 5 or 30 trials are different because it's a new state, it's a novel case,
Mhm.
it's a novel thing. I guess, would you have like 2 possible experiments where the question you can answer is like, what's the interaction between BLA and Gustatory Cortex in novel stimuli? Say, use those first 5 trials, let's say like over multiple days,
Yeah.
I guess, and then the second one is the after, like once the taste is not new anymore.
Right.
Like that, right?
Right. So I'm not entirely sure whether it's something that's GC and BLA specific. It can, so there's let me see if I can, oops, that's out of this. There's a paper by Francesca Schonberg. Yeah, I'm looking in 2 places. But basically, if you look at the attractor of the network, it just shifts in response to novel stimuli. And you could argue that, it doesn't really matter what network it is.
What do you mean by attractive?
So basically the equilibrium response of the network, right? What happens if you give a new stimulus, some Hebbian learning happens. The stable response of the network shifts in response to novel stimuli, and that will happen, I would argue, with every network, unless like it's hard code, right? If there's plasticity, you perturb a network consistently in a new way, it will shift. So I'm not sure what we'll look for in like a meaningful way between the GC BLA interaction in there. I'm sure things will change. I'm not quite sure what the hypothesis there is, if that makes sense.
Yeah, like it's not space.
Yes.
Space specific network, it could just be property
Of the network.
learning.
Yeah, exactly. That's what I think it is. Yeah.
I guess my second question is. Most, correct me if I'm wrong, most or all of this data was done in live animals,
Mhm.
like actual behavioral data.
Yeah.
How can you, or do you integrate, like computer coding simulation, like the more computational
Yeah.
stuff?
So I would love to try to make a model of the BLA GC interaction. I haven't done modeling.
Kind of the intention. We working with the main two of their lab to kind of make that.
Not a GC BLA model. And it would be so what Thomas will be taking up is for a long time, a quick tangent, and then I'll come back. Right. What I've shown you here is the assumption that these states are stable, right? That within a state, the response pattern is flat, right? It's consistent. But you could argue that there's intrastate dynamics, and we do find those. And so that's what we're trying to do with Thomas and Jonathan. But otherwise, there really isn't a model of GC and BLA that incorporates both the response dynamics and the learning. And coming from a physics background, I would love to do some model-driven hypothesis testing. But the closest model we have is Paul Miller's, which is very useful. I don't think it's an accurate description of the GC-BLA network. So I think there is a big gaping hole where a new model needs to come in, so that we can do more of that model-driven hypothesis testing. But I couldn't tell you exactly what that would look like.
But it is possible, like to make a computer model simulation.
For sure, right? It's and it's very much needed. Someone just needs to do it. Cool. Well, thank you so much, everyone, for your attention. If you have any questions afterwards, please feel free to email me. As I said, I love speculating. So I love discussing random things. But thank you for joining.